speakers here.
The speakers here, okay.
All right, I'm really delighted to have Dr. Mark Levin here. Mark really joined us how many months ago?
In January.
And he came to the Division of Pediatric Cardiology, Department of Pediatrics, from Children's Hospital in Philadelphia. Before that, he did his residency at Columbia University in New York, and before that his MD at Boston U in Boston. And he has done some very interesting research at the cellular level. And Mark, well, first of all, welcome to Washington University, even though it's a few months. But welcome to our seminar. And Mark will tell us about triggers of atrial fibrillation from the pulmonary veins. I think you all know that this is one of the hottest topics in cardiac electrophysiology. So.
Well, thank you very much. Really appreciate being invited here. It's a little intimidating, honestly, bringing the coals to Newcastle. So please feel free to add or correct what you'd like. And really thank you for welcoming me.
Can you try to speak up a little? Because
sure.
There's supposed to be a working and operational audio system here, but it hasn't been operational in a long time.
Okay. Well, please let me know if I'm not speaking clearly or loudly enough. Thanks very much for inviting me, and please let me know if there's anything I could make more clear. So what I hope to talk to you about today and get some feedback from you about relates to a novel model of pulmonary vein triggers using a mouse model. So why study atrial fibrillation? It's really the most common arrhythmia for necessitating admission. It affects a huge number of people, 2.5 million people in the U.S. alone, and as the population ages, that number will continue to balloon to people project at least 10 million people by 2050. It's a major source of morbidity and mortality. Mostly this is related to thrombus formation. So atrial fibrillation is really characterized by an irregular rhythm. This leads to discoordinated contraction between the atria and the ventricle. So there's stasis and thrombus formation. This thrombus can then become dislodged and go to the brain where it will cause a stroke, or it can go to the coronaries where it can cause myocardial infarction and lead to death. It's really a major drain on health dollars, and that's also a particularly important topic in these days. The current therapies are really not great. There are antiarrhythmic therapies, which, depending on what the literature says, between 20 and 60% effective. They're invasive approaches, and I apologize, Dr. Schussler, I should put surgery up there as well. Really one of the pioneers of surgery for atrial fibrillation was done by Dr. Schussler and Dr. Cox here, and that's, I'm not sure what the numbers are for that, honestly, but had a fair number of patients who've had that is efficacious. Catheter ablation involving pulmonary vein isolation is between 50 and 90% effective. This procedure carries risks, including risks of death and risks of perforating the posterior atrium, and oftentimes these pulmonary veins can reconnect. Ultimately, it doesn't address the underlying cause. It simply aims to disconnect the pulmonary veins from the remainder of the heart. Really what's needed are better animal models. So prevailing wisdom about different models of atrial fibrillation are summarized here, and I should point out that atrial fibrillation is really a multifactorial disease, and there are multiple different forms, and even within the same patient it can take different forms. At one end of the spectrum, there's the multiple reentrant type of circuits, which is more characterized by a chronic atrial fibrillation. There are shortened refractory periods and tissue heterogeneities, and based on this, there are multiple wavelets that reenter and perpetuate themselves. And at the other end of the spectrum, particularly in the last 10 or so years, there's been a lot of attention paid towards ectopic foci, mainly coming from the pulmonary veins, and these foci drive atrial fibrillation in the remainder of the heart. This type of atrial fibrillation is sometimes known as lone atrial fibrillation, and it's more often characterized in structurally normal hearts. A lot of the work over the last 10, 15 years has been made in understanding substrate remodeling and contribution of substrate abnormalities in perpetuating atrial fibrillation. And these include understanding of atrial fibrosis changes, as well as changes in tissue refractoriness or remodeling of ion channels, as well as understanding calcium handling defects. But really there hasn't been a lot of work or a lot of increased understanding with respect to the pulmonary vein triggers. This is really one of the main inspirations for why people have been looking towards the pulmonary veins. Miguel Hasseguer in 1998 published this article, and what he was able to show was that he could record pulmonary vein drivers using invasive catheters, and by recording those, he was able to demonstrate that abnormal electrical activity, and then disconnect the pulmonary veins from the remainder of the atria, and show that this led to a cessation of atrial fibrillation. But really the underlying mechanisms that drove these triggers remain unknown. Well, what I hope to talk to you about today might help explain some of these observations. I'm going to talk to you about protein I've been studying is doped chromatomerase, and this is a mouse that I'll describe in a little detail. In place of the actual protein, we can engineer a mouse that reports the location of this protein after we develop it with this blue dye. And what you see here is an explanted lung and heart from a mouse that has this gene which reports the presence of this protein. And you can see really distinct punctate staining within the pulmonary veins and the posterior atrium of this mouse. Furthermore, when you inactivate this gene, you reliably get atrial fibrillation in a mouse. And I'll draw your attention to the surface electrocardiogram. And AFib is really characterized by an irregular rhythm, and you can see that by these intermittent ventricular electrograms in response to the rapid pulmonary contractions and depolarizations. Here are the invasive electrocardiograms, right atrium and right ventricle. Here's the right ventricular recording, and you can see these are ventricular depolarizations, and interspersed between those ventricular depolarizations are very rapid multiple atrial depolarizations. And by contrast, you can look at the end of this. This is sinus rhythm, and by contrast, you can see an atrial depolarization followed by a time delay, followed by ventricular depolarization, followed by another time delay. So first, what I'll talk to you about relates to the temporal and spatial distribution of these cells, and some of the protein and mRNA expression of these cells. Then I'll go through some of the in vivo whole animal data, followed by some of the in vitro data, and hopefully I'll get time to tell you about the genetic ablation as well. So the enzyme I mentioned is doped chromatomerase. It's an unusual protein. It's involved in melanin synthesis, and it complexes with two other enzymes, tyrosinase and tyrosinase-related protein 1, in order to help produce melanin. It's the downstream target of several transcription factors involving PAX3 and MITF, which are, MITF is sort of a master regulator of melanocytes. DCT itself marks melanoblasts, which is an immature form of a melanocyte and doesn't always contain melanin itself. It's also been found in the brain. And one unique feature of this protein is that it contains copper binding domains. And really might be because of these motifs that allow it some of its unique features with respect to calcium binding and perhaps reactive oxygen species handling. going to bring this up. I'm going to mention this mouse a few times and another related mouse. And again, we take a fragment of the promoter, which is a regulatory sequence for a gene, and replace the downstream area where protein would usually be expressed. We put this lac-C protein. And this transgene is then inserted, and if we're lucky, it gets incorporated into the genome, and you can use this as a tool to see the localization of your gene of interest, which is included in some of these pictures here. I'm not sure if that's really projecting very well, or if it's very clear. This first photomicrograph on the left is it's an in situ hybridization. So what you can see here, here's the apex of the heart. Here's the posterior portion of the heart. Here's the region of the AV node is about here. The right ventricular affluent tract and the aorta. And what we're seeing here is labeled to messenger RNA that encodes double chromatotomlase. And that's really in the area of the posterior atrium and the pulmonary veins, as well as in the area of the AV node, and some within the affluent tract. The next three pictures show our whole mount samples using that mouse that I described for you, where in the place of the gene, we have that enzyme, beta-galactosidase, which we can then develop with this enzyme. And here you're looking at the posterior portion of the heart. From behind, looking forward, this is the posterior atrium, the right atrium, the left atrium, tricuspid valve, mitral valve, and here's the AV annulus. And really you see a tremendous amount of staining in the posterior atrium as well as in the AV annulus. This is the septal surface of the right ventricle and the right atrium, and landmarks include the inferior vena cava, the coronary sinus, the foramen ovale, and really again really distinct punctate staining in this region as well around the area of the coronary sinus, which is also a common area where one sees triggered activity arising. And this is that same picture I showed you at the beginning.
Yes, that's a question.
Sure.
Methodologically, you made a transgene with the promoter driving the beta-galactosidase.
Correct.
So then it's presumably expressed on a wild-type background. So
Correct.
You have normal DCT and you don't have arrhythmias in these mice.
Exactly.
And then that says DCT CRE R26R.
Right.
What's going on there?
So this is a little different method that instead of using the transgene, before we made the transgene, we used the DCT mouse, which is a CRE knock-in mouse. And then there's another reporter mouse that you can use, which is an R26R mouse. That allows you to really fate map a gene. And what you can do for that, it's a constitutively active promoter, meaning it'll express in every cell. And there's a stop codon just prior to a reporter beta-galactosidase. In the presence of CRE, that'll be excised, and it'll report. It basically reports something very similar.
I see. So this is a DCT promoter-driven CRE.
That's what this is. Yeah. I was trying to simplify a little bit by not going through that. I should have looked through that as well. Is that?
So excuse me.
Yeah.
Does those stain correspond to any structural feature in the tissue? Or?
Well, it does correspond to a subpopulation of cells. So before, I'm not sure exactly. I was curious what Dr. Efimov might think. I'm not sure if he's seen these, but I would say this is roughly in the area of. It's very superficial, and it's probably. It's more superficial than the compact node. I'm not sure if he would think this is in the area of the fast pathways or not. Does that help? Is that clear? Is that the question?
Yeah.
So it could be near the node in the atrium.
It's certainly not deep enough as the compact node, but it's certainly in the area of the node.
And then the ventricular staining was in one particular area of the ventricle. What's
Well,
that
really,
correspond to?
so here's a lower power. I'm not sure if that's clear enough, but really, it's very minimally expressed in the ventricle itself. This is the tricuspid valve, septal leaflet. It really doesn't extend into the ventricle myocardium itself. But
Is that a hot spot for VTAC?
yeah, so there is, a lot of people will talk about, actually the speaker that was here a couple of weeks ago, Dr. Josephson, will talk about the ring of fire. And there are a lot of hot areas, particularly underneath the leaflet, valve leaflets themselves. There'll be areas which trigger arrhythmia, both under the valve leaflets and in the area of the coronary sinus, and sometimes also in the area of the SVC as well. So we got 8 human samples, and we were able to demonstrate in human tissue that these cells were also expressed in human tissue. We found expression in pulmonary veins within the intima, within the media, as well as in the adventitial layers. Here on the left, you can see staining in the intimal region, and here this is my left. On my right, you can see co-staining with both DCT as well as for the tyrosine kinase receptor KIT.
So this is human tissue for what? From what? Can you say something more about the patients?
Well, we weren't allowed to get a lot of. There was certain barriers to know what a lot of clinical information, but these were adults who, for whatever they were normal, but they wouldn't use these hearts for transplants.
Okay.
Now the transplants, they have very stringent requirements for what they'll accept for.
But they were classified as normal, but
Yeah.
Okay.
Yeah.
Not transplantable.
They're normal, but they're not transplantable, so it's a little counterintuitive to me. But one of the kids was, well, I don't want to violate
Okay,
him,
that's
but
fine.
person was in an accident. So first we went through the distribution, and I think I went through that with you. Next, what we wanted to do was see if these cells expressed any other defining proteins which are characteristic of any other particular cell lines. First we were quite surprised to find what's commonly thought of as a melanin synthesis protein in the heart. And we wanted to see if these cells expressed other related proteins, including MITF, which again is a transcription factor for melanocytes, tyrosinase. PAX3 is usually found in these cells as well, and tyrosinase related protein 1. What you can see here on the left is staining using the dopachrome tautomerase antibody. This is mouse tissue in the heart. In the middle, you can see the co-staining using these other antibodies. And the third column is the merge. These latter two proteins we found at later time points, after E16 or E17. Well, we also wanted to see if there were commonly known proteins expressed in these cells, including those that are typically found in myocytes or in neurons, or fibroblasts, or neural crest, endothelial cells, secondary heart field, and we didn't see expression of any of these commonly found proteins within these cells. This is a complicated experiment, and again, please correct me if it's not completely clear, but what we were able to do was do transcriptional analysis of several different cell types. One cell type was these DCT expressing cells in the heart. Second cell type were cardiac myocytes, and the third were dermal melanocytes. And really we wanted to see in what ways were these cells like atrial myocytes, and in what ways were these cells like dermal melanocytes. So we used these methods really pioneered by Jim Eberwein, who was the first to do linear amplification of mRNA, and we extracted mRNA from 15 different cells and amplified those. As typical, those genes encoded in red were expressed greater than twofold above the mean. Yellow is roughly the mean, and blue demonstrates twofold below the mean. And in a non-biased way, we asked in what way were these cells like each other, and in what way were they dissimilar to each other, using cluster analysis and a Pearson's correlation. Each one of the cell types segregated to each other, so that validated our method in that all the DCT expressing cells segregated with DCT expressing cells, all the myocytes segregated with themselves, and all the dermal melanocytes segregated with themselves. What was somewhat surprising to us was that using this analysis, we saw that the DCT expressing cells were more similar to cardiac myocytes than they were to dermal melanocytes, which we weren't anticipating. We were also able to demonstrate that these cells at least expressed message of several important proteins that were necessary for cell excitability. And those included potassium channels as well as calcium channels and sodium channels. Using the DCT LAC-C mouse, we were able to use that LAC-C precipitate to identify these cells by electron microscopy. So the same thing that gives you that blue color will give you an electron-dense precipitate that you can identify by electron microscopy. And that precipitate, you can see these spindle-like areas dispersed through here. And what was several striking things that we found when we looked at these cells. One, where there were multiple caveolae along the cell membrane of these cells, which are typically found or correlated in calcium handling or reactive oxygen species handling types of functions. We also found some, but not all, of the DCT expressing cells express melanosomes, which are the organelle that contain melanin. And this one cell here has type 3, which are not completely mature melanosomes, and type 4. The abutting cell doesn't have any melanosomes.
No contractile?
No, there was contractile machinery, but what was surprising, they weren't the same type of filaments. It certainly didn't look the same. There was no sarcoplasmic reticulum either, which was another really surprising, or
Yeah, go on.
which was another interesting finding for us.
Do you see any evidence for gap junctions with myocytes?
So that's what really we were looking for. That's the reason we did this experiment. And honestly, there were some things that looked nothing convincing enough for me to publish. The technique is very difficult, and it's much, even to get through one particular cell, you have to really be lining up at the right plane at the right time and place to really do it. I think I'll be able to show it in a better way, but I never did it to the level that I thought was publish worthy. But that's really the impetus behind that. You remind me though, with the transcriptional analysis though, as you probably know, the connexin antibodies are not great, and we've got clear negative staining for connexin 40. We didn't get the connexin 45 antibody to work initially, but we saw a message using that transcriptional analysis. And then we went back later and got another antibody from someone else, and we did find and did show staining for that connexin 45 antibody. And I'll show you later, we did a lucifer dye transfer study, which showed that there's dye transfer. So to look more into the physiology of these animals and really try and understand what function does this enzyme or these cells play in the mouse heart, we were able to get this mouse from Dr. Friedrich Biermann, who's a scientist in Switzerland who knocked this gene out. He put Cre into the first exon, and really the only functional difference he saw was there maybe a slightly diluted coat color. These mice were viable and really had no other abnormality that were obvious. first question we really wanted to see was, when you knock this gene out, were these cells still present? And by mating both the DCT lac-C mice with the DCT Cre mice, we were able to address that question. So on the left, we have the control DCT heterozygote mouse. He's got one DCT normal allele and one knocked out allele, in addition to the lac-C transgene. And in the posterior portion of the heart, you can see some staining as predicted. In the DCT null mouse, again, we still see, I'm sorry it doesn't project as nicely as I wish, but you can still see positive lac-C staining in the posterior atrium. We then wanted to look at the function of the hearts and make sure that the ventricular and atrial function was normal, and we were able to demonstrate that quantitatively in the ventricle. There are really no significant differences in any of these measures, and I have some images showing this is an M mode through the short axis of the knockout mouse as well as the control mouse, showing normal contraction. Really, for the atria, there's no quantitative measure of atrial function, but you can infer that somewhat from the ventricular function. And you can also see qualitatively, again, here's the DCT null mouse, and here's the DCT control mouse. In diastole, you can see the atria relaxed, and in systole, it's contracting, and here's the control on the other side. Well, next we did invasive electrophysiology studies, and these are done analogous to the way that they're done in humans. And the types of information you can gather from these types of studies include baseline measurements of conduction through portions of the heart, as well as refractory periods. And the refractory periods are basically measures of action potential duration. They're really a stand-in for action potential duration. And the second group of tests you can do give you a sense for how prone a particular tissue type or person is to an atrial arrhythmia or a ventricular arrhythmia. And you do this in a progressively more aggressive way to try and elicit arrhythmia. Here's an example of the setup we use. It's a 1.1 French catheter, octopolar catheter. There are two pairs of electrodes in the distal portion of the electrode and two pairs in the proximal portion. So if you lightly anesthetize the animal, you put some subcutaneous ECG leads, which allow us 6 different leads. We do this under the microscope and do an IJ cut down. If you position the catheter correctly, you can get both ventricular electrograms as well as a His electrogram and an atrial electrogram. Here we have this is an old Bloom stimulator which we use to provoke arrhythmia, and we had an old human setup which allowed us to record and analyze the information. Well, when we did this in these animals, none of the measurements in terms of conduction through the atria, through the His, through the ventricle, or any of the refractory periods were different in the control when compared to the DCT null animals. What we did find a difference in, however, was a number of atrial arrhythmia episodes between the DCT null mice and the control mice. The DCT null mice had 27 episodes of atrial fibrillation or atrial tachycardia, and the control had 2. The 2 episodes in the control mouse were 174 milliseconds and 540 milliseconds. The average for the DCT null mice were roughly 781 milliseconds. Cycle length was quite different between the 2 groups. The cycle length in the DCT null were really quite fast. 9 of 11 of the null mice had the arrhythmia, whereas only 1 of the DCT control mice had that.
Didn't you show us, oh, okay, I was going to say at the beginning, didn't you show us that they had spontaneous atrial arrhythmias?
I'll show you that as well.
Atrial tachycardia.
Yeah, they did. That's the next slide.
I've shown you this, so I'll jump to the refractory periods were the same. This is spontaneous. So we implant telemetry monitors into these mice and let them recover for a couple days. What you can see here is the way we reasoned that it was atrial tachycardia or atrial fibrillation in this case, the P wave morphology and the P wave axis. So here the P waves are upright and have one morphology, and here the morphology and axis is really different. And you have this irregular pattern of ventricular depolarizations.
Did this just occur spontaneously on a recording, or you didn't do anything to provoke it?
No, we just left them overnight and let them run around and came back the next morning. And
do these animals show like premature beats too?
Well,
Atrial ectopy kind of things?
I didn't see it. It's basically I have to look at every single waveform. So it's possible that they had those. It'd be really hard for me to see it. It was hard enough for me to see this by literally like looking through every single waveform. But does that answer your?
Yeah. With time, does the occurrences of AFib or atrial tachycardia progress, and do they become more frequent?
You know,
or more
we only did this.
duration?
Well, I get it's a very good question. And the one sort of teaching point about atrial fibrillation that people commonly invoke is that atrial fibrillation begets atrial fibrillation, and this would be one way to sort of look and evaluate for that. I'm not sure is the short answer. We just looked at a one time point at one time, and this is what we saw.
This looks different than the induced because the speed within the atria looks to be varying. Some of them have a lot of rapid beats, like the first induction after the arrow looks like 3 or 4 very fast atrial beats, and there's 5 or 6 very fast ones a bit later on, and then it slows down in the middle. Whereas in the induced ones, it looked like it hit a very concrete rhythm. Or were the induced ones stimulated? Is that what those
so the
are?
induced ones were stimulated.
At
So
that
we do this in
frequency?
burst pacing.
Oh, okay. So that's the pacing rate.
This is, so you give a train, there's a different sequence for different parts, but essentially the way this was done, you give, it's 20 impulses at 50 milliseconds, and then the last three beats that you deliver are even shorter. The other difference between this and the spontaneous is obviously anesthesia, and really you'll have different properties in AV node conduction based on anesthesia and catechols and endogenous catechols, as well as what we're changing.
Can you go back one slide? That one. So here, the 250 millisecond bar shows 3 beats. So it's 3 times 4, so it's 10 to 12 beats per second. So that's a heart rate of 600 something. And so, which is normal for a mouse.
Right.
And then when it goes into AFib, it's clearly slower.
Right.
So is that, I mean, in people, it's the other way around.
Well, not always though. I mean, you have. I mean, the atria is going very quickly, and the AV node will not. The AV node is designed to filter atrial impulses, and there will not be one-to-one conduction. And sometimes in a somewhat paradoxical way, it'll look slower. I don't think that's that uncharacteristic.
I'm just asking, is mouse AFib usually slower than mouse sinus?
That's a good question.
This is a non-sedated mouse.
This is a non-sedated mouse. So
it's as close as you can get to actual physiology.
yeah. Did that answer well enough your question?
Well, you know that the AV node can respond fast enough to go at 600.
Right.
And so, do you actually count the AF rate here? It looks slower, at least from where I'm sitting. I'm just asking, I don't know if there's a mouse.
No, this is tachycardia, and the atrial tachycardic rate here is faster than the spontaneous rate, right? Than the normal sinus rate.
The ventricular rate is clearly slower.
Yeah.
The atrial rate is clearly
Faster.
faster.
Yeah.
Now, in patients and in people, that's something I've seen. In kids with atrial flutter, there's a very fast flutter rate, but the ventricular response is not necessarily a tachycardia rate. So
I think generally in humans we, or at least in adults, we see a faster ventricular rate with a faster atrial rate. Flutter being the exception where you sometimes have 4-to-1 block or something, but generally and to say that's definitely flutter.
oh, I wouldn't have said that's flutter. It may or may not be.
But with AFib, I think we would be more likely to see a faster ventricular rate. So I think that's Kovac's question.
Yeah.
Well, but it's a mouse, and it works at
Right.
600, all the time. So I wouldn't make extrapolations from this to what you see humans to do.
And this isn't fibrillation, is it? I mean, that's still.
I'm not sure what. I'm not sure what people can argue. I would say it's certainly a spontaneous atrial arrhythmia. It's certainly. I think it's very likely to be atrial fibrillation. If someone wanted to dissuade me otherwise, I would entertain discussions. The other part of this is that a lot of noise. These are pretty noisy apparatus, and it's not easy to get a great signal. Certainly, some of this looks very fibrillatory to me.
Mm-hmm.
Some of it does and doesn't, and it's hard for me to know with 100% certainty that it's definitely fib. But it's clearly not sinus. It's clearly a spontaneous arrhythmia, which is unusual in a mouse model. I mean, for a long time, people thought that because of the wavelength of the arrhythmia, people predicted that you wouldn't even be able to fibrillate an atrial, a mouse atria. So it's something. I think it's real, and it seems to correlate with some other things we found. I'm not sure how much I can generalize from this or how much to read into it, but I do think it clearly is a spontaneous arrhythmia. And
Is there any difference in the size of the atria between the control and this group?
no, again, we looked for that, but we didn't see it either grossly by histology or by echo.
So there was no difference. There's no dilatation.
No. Here's the, it's hard to get a quantitative, it's hard to get a quantitative function for an atria. For a little bit, we thought about trying to get MRIs on these, but even with the MRI, it's not a regular shape that you can really calculate something with. But from what we could see, from what we could gather from a number of different, yeah.
Mark, did you study any of them in vitro, where you
Yeah.
would have a much better resolution and be able to see exactly what kind of electrical activity there was, and maybe even map the atria?
Well, so we didn't do, I mean, it'd be very interesting to do like what Igor does. No, we haven't done that, and I've been hoping to get the mice here so we could do something like that here. What I will show you is, and maybe skip ahead to that, is that we did do some some in vitro work. We explanted these hearts, and we were able to grow these cells in culture and identify, similar to the way we identify these cells with the beta-galactosidase, we saw these cells with epifluorescence. You can see, here's an example. And what we did see, we did whole cell current clamp recordings and found that the action potential duration in the DCT null cells was at least twice that of the control cells. And the atriomyocyte action potential durations were roughly equivalent between them. This was by ANOVA. So really all of the DCT null, this is cardiac melanocyte-like cell or DCT expressing cell, really had profoundly prolonged action potential durations compared to the atrial myocytes.
Did they have any spontaneous?
Yeah, they did. So, so this is a spontaneous, spontaneous recording, and like what Dr. Rudy had described at least 10 years ago, there's this pause prior to that, and then the rise from the third phase of action potential. And we saw this consistently, and we never saw that in the control. So we did, here's the Lucifer dye transfer studies that I mentioned to you. Again, Lucifer dye it's a small polar molecule, and it will pass through gap junctions. And functionally, it's a way to demonstrate that cell is electrically coupled using gap junctions. So we got in the cell attached mode, we got whole cell, and the dye transferred from the one cell to a neighboring myocyte.
Are these cultured myocytes?
These are cultured. These are cultured.
3 days?
This is 2 days. We also, using the same preparation, we did some calcium imaging using calcium-sensitive dye, flow-4, and saw correlating types of phenomena. On the left, this is the DCT control heterozygote mouse. On the right is the DCT null. And again, I'm sorry that this, not sure how well it's projecting, but there's a myocyte, and then using epifluorescence, next to it there's a DCT expressing cell, or a CMLC. And using flow-4, we were able to measure calcium flux as correlated by changes in fluorescence. Here's the fluorescence change within the DCT expressing cell, and here's the fluorescence change in the myocyte, and that looks pretty consistent. In the DCT null cell, by contrast, again, we see this pause followed by what may be an EAD.
This is an EED in the fluorescence.
Yeah.
Interesting. And the pause is about 2.5 seconds, I guess.
Yeah. So to sum up this part so far, again, we saw very reliable in vivo what seemed to be atrial fibrillation, both induced as well as spontaneous. In vitro, what we saw was very clear action potential prolongation in the DCT null cardiac melanocyte, which we didn't see in the control DCT melanocyte, and the action potential duration of both myocytes were roughly equivalent. And we saw these EADs during spontaneous recording. The next set of experiments we undertook were to see, well, in the absence of these cells, did we still see arrhythmia? And we tried to go about that a couple of different ways. This was the way that we ultimately used. There are there are mice which have spontaneous mutations and don't have pigmentation. And one of those mice is the WWV mouse, which has several different spontaneous mutations in the tyrosine kinase receptor KIT. These mice lack dermal pigmentation, so we thought there would be a decent chance that they would lack pigmentation within the cells in the heart, and that's actually what we found. Here on the top is the control mouse, and this is the septal surface of the right ventricle in the area of the AV node. You can see a lot of pigmentation. This is a valve leaflet and posterior atrium, pretty obvious. In the WWV mouse, you don't see similar pigmentation. We grew these mice up, and we performed similar electrophysiology tests, like I mentioned, for the provocative invasive EP studies. And we found that the WWV mice had similar amounts of arrhythmia as the control mice. The next step, what we wanted to do was what happened when we crossed these mice onto a DCT null background. So was it the absence of the cells, or was it the absence of DCT, or that really caused the arrhythmia? Well, similarly, when in this method of the DCT null mouse, on the background where there are no pigment-expressing cells, again, there was roughly comparable arrhythmia as in the control mouse. Well, when we looked at the DCT null parts by EM, we were struck by a couple of things, and one of those things were abnormalities of the mitochondria. And these abnormalities are consistent with what other people have described as mitochondrial injury secondary to reactive oxygen species. And that's characterized by somewhat dilated mitochondria and cisternae, which are enlarged. And we followed up on that, and we wanted to know if we fed mice, the DCT null mice, which are the mice that had arrhythmia, if we fed them this scavenger for reactive oxygen species, would that restore some of the function back to normal mice, and would the arrhythmia go away? And we were able to demonstrate that to some degree. We fed these mice temple, which is a raw scavenger, for about a week, and studied these mice. And again, we didn't take away all the arrhythmia, but certainly less than their litter mates who did not receive any of the temple. So in summary, we described a group of cells that populate the pulmonary veins and the AV annulus. These cells certainly have transcript necessary for calcium handling and cell excitability. The mouse, intact mouse, shows the type of disease that's similar to what we see in atrial fibrillation in a structurally normal heart. When we did in vitro studies, we were able to demonstrate that these cells, the DCT null cells, had action potential prolongation and spontaneous EADs, as well as frequent calcium oscillations. And when we ablated these cells, there was diminished atrial arrhythmia. So now I've been working in Dr. Narbonne's lab, and we're going to pursue some of these things further. And really, some of the big questions that we have are, are these cells really in humans, and does this really, is this really relevant to human atrial fibrillation? And another major question is, what is the mechanism through which arrhythmia arises? Why is there action potential prolongation? And those are some of the things we hope to uncover. So thanks for your attention. These are a lot of people that have helped back in Philadelphia, and I appreciate your attention.
So you showed that those cells have various ion channels. How did you do that?
So again, it's a very clever technique devised by Jim Eberwein, and the technique involves linear amplification of messenger RNA. You get whole cell on a cell, and essentially what they do is they draw up the contents of the cell into the pipette, and in the pipette you have everything you need to amplify the message. So you do a couple rounds of amplification actually in the pipette itself, then you extract that and you amplify further and do microarray analysis on it.
So you can do it in the DCT null cell,
Yeah.
and do you see?
Yeah, we haven't done that yet,
Okay.
but will.
So it's a post hoc analysis. You have to do the patch clamp, and then you take the pipette and analyze what's in it.
Yeah, that's the way it was originally. The way we did it, we didn't patch on it was a separate group of cells that we patched. It's actually a much lower resistance pipette, and you really bring up the whole. You can do it that way, and people have described experiments where they recorded and then they drew up the contents. This we really just wanted to get as much as we could.
Did you do any coarse pharmacology to see whether if there's a K current missing that gives you the?
We didn't do no. We did some very preliminary work on that, and that's really why I came here. Why I wanted to work with Dr. Narbonne, and really, I mean, obviously, potassium channels are really an obvious target for what to look at, and I think it's a reasonable place to start.
In your echo assessment, did you do transmitral Doppler?
You know, they didn't. The machine that they had, we couldn't do. It was the version. I can't remember They didn't have the Doppler package to do that. It didn't make sense to me. But they didn't. We weren't able to do the color Doppler analysis. Is that what you're referring to? To do Doppler inflow of the mitral valve?
Well, I mean, although mouse hops are very fast, and normally you would, once the E waves and the A waves separate,
Yeah.
then if you have a Doppler data available, especially when you anesthetize the mouse,
Yeah.
the absence of the A wave is pretty clear that the atrium is not contracting in a functional way, and then especially if you have transient arrhythmias,
Yeah.
then presumably you can see the A waves come and go.
Yeah, we do have some data like that, and actually maybe I'll show you that data, because I'm not an echo person, honestly, and we did. The person who did the echoes did Doppler inflow. It did appear that how did he describe it? You can see. He didn't think that there was any difference, but in my, there was something about the E to A, ratio that didn't make sense to me, honestly.
So there are two things that I'm just trying to make at the time.
Yeah.
So the reason there is an atrial kick,
Yeah.
as opposed to everybody in AFib, at least as far as Brown Wall showed in the 60s, was that DPDP in the subsequent ventricular contraction is steeper. The atrial kick stretches the ventricle, it preloads it, and you get an increased DPDT. So if you have, in this kind of an experimental setup, if you can get a molar wire into the mouse ventricle, you can actually plot DPDT, you can compute DPDT as the atrial things come and go, and even if you don't have clear atrial function data, as you see the peak positive DPDT in the ventricle go up and down with the atrial stuff, it's very compelling physiologic evidence that the atrium is not organized. It's not making an A wave to pre-stretch the ventricle to increase the DPDT. So that's one way. The other way is if the heart rate is slow enough. So in the mouse, what happens is instead of the ventricle sucking the blood in and then diastasis, atrial contraction, I'm going to try to get my hands to work right. So it goes like this. So the atrium and the ventricle work together, and atrial systole and ventricular suction are timed so that when the ventricle sucks, the atrial pushes simultaneously. You can separate those two with anesthesia. You can see the E and A waves literally separate on echo as the heart rate slows. So all I'm trying to say is that there are physiologic ways to assess the presence or absence of organized atrial contraction.
So to define the atrial fibrillation better. I mean, honestly, when we did the echo studies relatively early in the process, and we weren't expecting to see spontaneous AFib. We were very surprised to see it, and actually the PI asked me why I was wasting my time even trying to do that experiment, because no one expected to see the spontaneous. But I'll show you the waveforms, because it did look somewhat like what you're describing, but I'll show you. I'll send you the video.
Do people with melanin deficiencies show any evidence of arrhythmia?
It's really funny. So So one of my really good friends. So I didn't show you, but there are a bunch of different genetic abnormalities with different dermal abnormalities, and one is the splotch mouse, and there's a flash of pigment of white pigment loss right here. So one of my friends has that same mark, and he has AFib, and he's a relatively young guy. So nothing has been reported, but there's certainly anecdotal evidence with that. It goes along with like a SOX10 mutation, but nothing's been described officially.
I noticed that your atrial tract cardiac cycle length drops in the normals. What do you make of that?
The cycle length of in tachycardia
Yeah. Because I would think cycle length, once you're an AFib, should be a property of the atrial myocardium. Unless I'm wrong.
Well, if it's a completely focal mechanism, that suggests that this is a truly focal mechanism, and that your observations on the EAD, if it's independent of the action potential duration or refractory period, that would suggest that it's a, and your data showed that refractory periods were exactly the same, so that makes me very suspicious that this is truly a focal tachycardia. It's not dependent on this, it seems to be totally independent, which is very interesting observation.
It's very hard to get re-entry in this small atrial curve.
Yeah.
But remember, if the atria are not dilated and you have such a small atrium in the mouse, it's probably mostly due to ectopic focal activity rather than re-entry. There's another question from your slides. It's interesting to see the calcium abnormalities.
Yeah.
But it's not really clear. Do you have a hypothesis what causes what? It's very hard to separate, because from what we showed, it could be that the abnormal electrophysiology is causing it, because there were very couple.
Well,
I've never seen an EAD. It's hard to talk about an EAD in the mouse action potential, but I've never seen an EAD in fluorescence calcium.
Yeah.
Which to me suggests that it's.
Are you saying that EADs are not associated with
it's probably secondary release because of the second action potential.
Yeah.
That's probably what it is, not the other way around.
My understanding was that the action potential prolongation is likely to lead to the calcium abnormalities, or at least sufficient time for the calcium inactivation to recover from inactivation.
Yeah, but that started. Your EAD really started from very low voltages. It was very low. It was at the end of the action potential almost. So that's not the mechanism.
That's not the mechanism.
Can't go. It's below the voltage for calcium activation. So it's either sodium calcium exchanger or it's a whole new action potential that is causing a new release. But it's all interesting. I just don't know.
It's real, yeah.
It's definitely focal. It's all focal.
Yeah.
The EKG you showed, certainly looked AFib-ish. I'm just wondering, you said you looked through all that telemetry.
Yeah.
Did you see these pauses and then the EADs and then
see a lot.
AFib, or what was the pattern?
What
we were considering to be AFib,
So what I had to look through was basically what you saw, the very low resolution at top. I was looking at sheets and sheets that looked like you're looking through sheets that look like this, and basically when I could see a really clear rate change, that's when I would slow down and slow down. But it's very possible that there would be a single beat that would be off that I would clearly miss.
But there's no pause there. There's really no
right,
pause
yeah,
there.
well, yeah. The first, see that's AV, and then this doesn't look like an EAD, it just looks like it's chugging along, then all of a sudden you get atrial.
That's an extra.
Actually, that doesn't look like an expansion of what's above, because the ventricular rate is speeding up during the arrhythmia, it's not slowing down. Isn't it?
Pretty sure it was.
You have like 5 or 6 beats that look like they're normal, or 7 maybe. And then it,
I think this is right here. So the rate prior to this was even slower.
uh-huh.
These,
I'll,
again, I can pull it up for you.
Yeah, somebody there.
For the in vitro study, did you capture the cell from ventricular tissue?
I'm sorry?
Capture the cell from ventricular tissue?
Well, it was mostly atrial tissue. So, I really tried to. It's tough because if in culture, if it's too sparse, the cells won't live. So I couldn't just only take the DCT expressing cells. So I would take basically posterior atria and septum of the atria, and tried not to take ventricular myocytes.
So so there are like ion channel mutation that causes the atrial fibrillation, and now you have this. So can you give your take? What is the difference? What is the difference between those kind of ion channel mutation that cause the atrial fibrillation and this?
Well, again, it refers back to, is it really substrate or ion channel? In general, most of what's been described with respect to mutations involves shortening of action potential duration throughout myocardium, which enables or facilitates fibrillation. This is a very different mechanism. This is a group of cells that's initiating a trigger in the back of the atrium or underneath the valve leaflet or in the coronary sinus. And I'm not sure how much, I'm not sure if there is a correlated mechanism or gene or series of genes that are operational in the human, but that's obviously the key thing for me to look for. And do I imagine? I think